is vital for survival, and it protects us from infectious pathogens. For example, we can name some disorders of the immune system such as allergies and autoimmune diseases. So first we should know that we have two broad categories that immunity is divided into innate immunity and adaptive immunity. So first of all, before going to diseases of the immune system, we should know some of the concepts in immunology. So immunity is divided into innate immunity, which is called also natural immunity, and adaptive immunity, or acquired, or specific type of immunity. Innate immunity is always present. And what are the components of innate immunity? Epithelial barriers so the epithelial cells that cover our skin, different systems such as respiratory tract, gastrointestinal tract, urinary tract. These epithelial cells act as barriers against invaders. These invaders can be molecules from bacterial products or necrotic and damaged cells. So imagine these are cells that cover skin or intestinal mucosa. Then these invaders such as bacilli, they cannot enter the tissue because of having these epithelial cells. And there are also other cells that are important in innate immunity, including phagocytes, especially macrophages, and neutrophils. Phagocytes that they ingest different molecules and can kill them. So phagocytes and dendritic cells. Dendritic cells, the most important function is antigen presentation. Phagocytes also present antigen, but they can also phagocytose and kill them. But the main action of dendritic cells is binding antigens and presenting to other cells. Dendritic cells are present under skin, in different tissues, especially under epidermis. And a type of lymphocyte which is called natural killer cell. Natural killer cell, NK cell, is also important in innate immunity. And some proteins such as components of the complement system. Complement proteins. So these are the components of innate immunity, including epithelial barriers, phagocytes, the most important of which are macrophages and neutrophils, dendritic cells, which the most important duty of which is antigen presentation, natural killer cells, and complement proteins. But what are the cells important in adaptive immunity. They are lymphocytes, including T lymphocytes and B lymphocytes. So, acquired or adaptive immunity is developed by lymphocytes, T and B lymphocytes. So these are not considered for innate immunity, but for adaptive immunity. So in this picture we see this part innate immunity and this part adaptive immunity. The epithelial barriers, the phagocytes, dendritic cells, phagocytes, which phagocytose ingest the microbes, the damaged cells in necrotic cells, components, and have the ability to kill them. Dendritic cells that their main action is to display antigen on their surface and present it to other cells, also to B and T cells. And natural killer cells are bound with antibodies, and these antibodies can bind antigens. And complements, proteins of the complement system, that one of their action is, as you remember, opsonization. For example, C3b, it covers this bacterium and can introduce this bacterium to special receptors on cells. This is innate immunity, and adaptive immunity, which includes B lymphocytes and T lymphocytes. This is the dendritic cell that has presented this antigen. This, imagine this is the microorganism. Then this microorganism inside the cell is processed, and some of its molecules, including peptides, are presented on the surface. So this round molecule on the surface of dendritic cell has developed from this microorganism. Is a peptide, for example, that after processing of this microorganism has been developed and traveled to the surface of dendritic cell and now is introduced to T lymphocyte. So this is the action of antigen presentation by dendritic cell. Then, by binding to T lymphocyte, these T lymphocytes are activated and changed to other types of cells with other names. And these are B lymphocytes that they have a special type of receptor called antibodies. So, T lymphocytes and B lymphocytes. These two cells have special receptors on the cell to bind antigens. The receptor of T lymphocyte is called T cell receptor. The receptor on B cell is called B cell receptor or antibody. So antibody is a receptor to bind antigen. This is an antibody that is bound to B cell, and its role is binding antigens. And this is a T cell receptor, a receptor on the T cell that binds antigens presented by phagocytes or dendritic cells. So this is the adaptive immune system. Now, about innate immunity, that innate immunity is always present and it is ready to defense against microbes and to eliminate damaged cells by presence of epithelial barriers that are present every time, and macrophages, phagocytes that are always present and they can defend against invaders. The important thing in innate immunity is to learn some new receptors that are important for binding different antigens. So this part, again, in this part we learn new molecules. So cells, including all the cells that play a role in innate immunity, they should have some special receptors that for the first time they sense or see the microbial products, and they have the ability to bind them. So, Giar, what is the main division of immunity? We have two types of immunity. Because you are speaking.
I'm studying. You have exam.
You are not studying, you're speaking.
Really, I'm studying.
But it's not time to study, it's not
You know,
time to listen.
Yes.
No time for studying.
Yes.
Studying is for home. Studying.
Yes, I know. I just repeat that because
You're repeating a lot. You are
no, we
just
discussed
speaking.
Okay, so sorry.
So
Innate, adaptive.
please listen. Now we have new things to learn again. We have 4 types of receptors that are the first receptors that they sense microbes and necrotic cells products for the first time. These are TLRs, or toll-like receptors. Nod-like receptors. Reed-like receptors. And lectins. So, these receptors that are present at different sites of a cell, being cell membrane, intracytoplasmic, or inside the vacuoles such as endosomes. What is the difference between an endosome and a lysosome? Can you explain what is the difference? What is the difference between an endosome and a lysosome?
No, lysosome contains enzymes, but endosome is just a 2-layer of phospholipids.
Yes, lysosomes have enzymes, hydrolytic enzymes for degenerating, for example, molecules. But lysosomes are only for accumulation of molecules that come inside the cell by endocytosis. They don't have enzymes. So these receptors can also be present on walls of endosomes. So these are toll-like receptors that can be present on cell membrane and also membranes of endosomes, vacuoles present inside the cell. And these are TLR or toll-like receptors. These are nod-like receptors that are present in cytoplasm. These are reed-like receptors and lectin. But they have different abilities. Lectin has the ability to bind polysaccharide of microbes. So it depends on the molecular structure. Lectins are able to bind polysaccharides of microbes. Nod-like receptors have the ability to bind different molecules, such as products of damaged cell. They can bind ATP released. So extracellular ATP can enter cell, and nod-like receptors can bind. Also, they can bind bacterial peptidoglycan. Reed-like receptors have the ability to bind viral RNA. So they can have antiviral effect, because by binding to viral RNA, some molecules are produced that will have antiviral effect. And TLR on the endosomes have the ability to bind nucleic acids of ingested microbes. So ingested microbes also have DNA, and TLR or RNA, and TLR of endosome can bind this nucleic acid. So these are 4 different types of receptors that they are the receptors that sense the different molecules of microbes, being carbohydrate, being proteins, being lipid. So with different structures are sensed by these different receptors for the first time. We also have other types of receptors, such as mannose receptors that bind to sugars of bacteria. So these are the different 4 types, and they have a name. These receptors are called pattern recognition receptors. They are called pattern recognition receptors that can bind antigens of microbes and damaged cells. And those antigens are called pathogen or damage associated molecular patterns, or damage associated molecular patterns. So antigens, if are from microorganisms such as bacteria, are called pathogen associated molecular patterns, PAM. Or if these antigens are from necrotic cells, are called damage associated molecular patterns, DAM. These are sensed by these different receptors called pattern recognition receptors. So we are studying immunology, not the diseases. This time we are reviewing the principal concepts of immunology. And then after this we will start diseases of the immune system. For the first time, these toll-like receptors were discovered in Drosophila, and the same name was given to mammalian similar receptors. And in mammals, we have 10 types of toll-like receptors. So, the concept of different receptors, and what is the result of innate immunity? Is inflammation. So cytokines and products of the complement system, they cause inflammation, they cause collection of inflammatory cells, and the result will be inflammation. Also, some molecules such as type 1 interferons have antiviral effect. So the result of innate immunity is inflammation and being antiviral. So by production of cytokines and activation of the complement system, the inflammatory cells will collect, and by molecules such as type 1 interferons, these are antiviral. Adaptive immunity consists of lymphocytes, I said. But again, adaptive immunity is divided into 2 types. Since we have B lymphocytes and T lymphocytes that are important in adaptive immunity, we have 2 types of adaptive immunity One is humoral immunity. That is developed by B cells. B cells have antibodies. Only B cells produce antibodies. Another name for antibody is immunoglobulin. Only B cells can produce antibodies. Antibodies are bound to B cells and have the role of a receptor, B cell receptor or antibody. So B cells, they cause humoral immunity, and this humoral immunity is for elimination of extracellular microbes. And another type of adaptive immunity is done by T cells, and they develop cellular immunity or cell-mediated immunity. So, cellular immunity or cell-mediated immunity is developed by T cells, and humoral immunity by B cells. And this cellular immunity is for elimination of intracellular microbes, and also tumor cells. So T cells and B cells produce cell-mediated immunity and humoral immunity for elimination of extracellular and intracellular microorganisms. So cells of the adaptive immune system are B cells and T cells. B cells are bound to antibodies. B cells can differentiate into plasma cells. And plasma cells, the difference of plasma cells with B cells is that the antibodies that they produce is released. But in B cells, the antibodies are bound to the B cells and have a role of receptor. So this is a B cell antibody is bound to the B cell, and this has the role of a receptor. This B cell is differentiated to plasma cell with an end nucleus, plasma cell. And this plasma cell role is production of antibodies. So these antibodies are not bound to plasma cell, but they are released and they do immune responses. So this is the difference between B cell and plasma cell. And plasma cell have the ability to synthesize a great amount of antibody in a little time. This is called humoral immunity of the adaptive type. So these antibodies can neutralize the microbe, neutralization of microbe, phagocytosis and complement activation. They have roles, these antibodies that are produced have these functions. We have three different types of T cells. So T cells are generally divided into three types. T cells. We have helper T cells, which T helper cells, cytotoxic T cells, and regulatory T cells. As the name shows, helper T cells, they help other cells such as B cells and other T cells for having immune responses. So T cells, they are divided into helper, cytotoxic, and regulatory. Helper T cells, they produce some different types of cytokines that these cytokines activate B cells and other T cells. Cytotoxic T cells produce some molecules that can directly kill infected cells. So this is the difference between cytotoxic and helper T cells. Cytotoxic T cells are killer cells, like natural killer cells. They can kill infected cells. But helper T cells don't kill the cells directly. And what is the action of regulatory T cells or reg T cells? They inhibit helper and cytotoxic T cells, and they suppress immune responses. So they have a negative effect on cytotoxic and helper T cells. They are for controlling immune responses. And helper T cells, as I said, they produce different types of cytokines, and according to this, helper T cells are divided into T helper 1 cells, T helper 2 cells, and T helper 17 cells. T helper 1 cells produce, for example, interferon gamma cytokine. T helper 2 cells produce interleukin 4 and interleukin 5. T helper 17 produce interleukin 17. So by production of different cytokines, different types of T helper cells have different functions. For example, T helper 2 cells play a role in allergies. So they have different functions. So T cells are divided into helpers, cytotoxic, and regulatory. Helper T cells themselves are divided into T helper 1, 2, and 17 subtypes. And regulatory T cells suppress immune response. So helper T lymphocytes, they have the ability to bind antigens presented by dendritic cells. Then they are activated and produce some cytokines. By production of these cytokines, they can activate T cells and B cells. They can accumulate inflammatory cells and cause inflammation, and they can activate macrophages by these cytokines. So these cytokines can activate macrophages, other T cells and also B cells, and also cause inflammation. So as you see, helper T cells don't kill infected cells directly. They cause activation of other inflammatory cells, other T cells and B cells, macrophages, and they cause inflammation. But cytotoxic T lymphocyte, which is also called CTL, when they bind infected cell, which can be a macrophage that has ingested a microbe, by releasing of some molecules through these cytotoxic T cells, they activate the enzyme inside this infected cell, and it will result in killing of infected cells. Regulatory T cells have
inhibitory role on T cells. They suppress immune response. So if we have an elevated or activated immune response by T cells, we should have presence of regulatory T cells to control them. So they should be present to have a balance in immune responses. Regulatory T cells suppress immune responses. And natural killer cells that have role in innate immunity, again, they can cause killing of bacterial infected cells like cytotoxic T cells. There are some receptors and markers on B cells, T cells, and phagocytes, antigen-presenting cells, in order to sense each other, in order to be able to bind antigens and present it to other cells. Every time you see that different types of receptors are needed, as we learn the presence of pattern recognition receptors, including which pattern recognition receptors are? What? What? This is the first time it's memorizing is difficult. So pattern recognition receptors include this picture. These are pattern recognition receptors. Toll-like receptors, lectin, NOD-like, RIG-like. These are called pattern recognition receptors that are important in innate immunity. They bind DAMP and PAMP that you said, that are antigens from damaged or necrotic cells or infection microbial products. So those are called DAMP and PAMP, but these are called pattern recognition receptors, including the general 4 types. So we learned these receptors, but we have also another types of receptors on antigen-presenting cells and T cells. So for the first time I said these 4 types of receptors are the first receptors that they see antigens. But there should be also other receptors that they can bind processed molecules of these antigens and again present them to the surface of the cell for binding to T cells. So these, for example, this is a T cell and this is a Toll-like receptor. Toll-like receptor, so it can be a macrophage. Imagine this is a macrophage and has a Toll-like receptor on cell membrane. This is a bacterium. One of the molecules of its cell wall, it's a protein, and this protein is an antigen that can bind this Toll-like receptor. So this is a bacterium, this is a protein that as an antigen can bind this Toll-like receptor. This causes activation of some signaling pathways inside the cell, produces some molecules such as cytokines that these cytokines cause activation of these macrophages and other cell types. So this is the first step. Then phagocytosis should be done. So this, by activation, this particle should be phagocytosed inside the cell. So then this is processed inside the cell, and some part of this microorganism, for example, this molecule, which is a peptide, again comes to the surface by another type of receptors on macrophages and different cells called MHC or HLA. So these pattern recognition receptors are the first receptors that can bind antigens. But then these antigens are processed inside the cell, and some molecules such as peptides, they appear again on the surface of the cell, and they are presented by a special kind of receptor called MHC molecules or HLA. Maybe you have heard that some patients that need bone marrow transplantation, for example, they should be HLA compatible with the donor. So this MHC or HLA is human leukocyte antigen, they should be compatible with each other. These MHC molecules present the antigen to T cell. This is a T cell. T cell that has T cell receptor. So now we want to learn MHC molecules that are receptors on these antigen presenting cells, and T cell receptor and B cell receptor, which are antibodies. These MHC molecules have two classes, 1 and 2. And they are expressed by chromosome 6, genes on chromosome 6. So again, after those pattern recognition receptors, we have newer receptors here for continuing of the immune response. MHC molecules, including class 1 and class 2, and T cell receptor present on T cell. Then these two receptors can relate to each other through this antigen. By binding, these T cells will be activated. So, this picture shows two cell membranes. This cell membrane is for a T cell, and this cell membrane is for phagocytes. So this is a cell, only its cell membrane is shown here, that has presented this peptide antigen to this T cell membrane by this MHC molecule. And this is the T cell receptor. So this is the antigen, these two are the receptors, important for binding the antigen. And this is CD4 and also CD8. I want to say that for helper T cells are CD4 positive, cytotoxic T cells are CD8 positive. Cytotoxic T cells have T cell receptor and a molecule called, a protein called CD8. T helper cells have T cell receptor and a protein called CD4. These CD8 and CD4 are called co-receptors.
they help T cell receptor for activation. So now we learn the concept of CD4 and CD8. A T cell cannot be both CD4 and CD8 positive. Some are CD4 positive, some are CD8 positive. CD4 cells are present on helper T cells. CD8 cell markers are present on cytotoxic T cells. So, for the first time that you are hearing these things, it's difficult, but it should be repeated and reviewed. And B lymphocytes, I said that they have B cell receptor, which is antibody. So this is the structure of antibody. This smaller part is light chain of antibody. This longer is the heavy chain of antibody. And they bind antigen. What are the tissues of the immune system? We divide tissues of the immune system in two parts, generative and peripheral. So we have some special tissues where all these reactions occur. Generative lymphoid organs, and secondary or peripheral. Generative is primary, peripheral is secondary. What are the generative lymphoid organs? Organs where for the first time T cells and B cells are developed and mature. They include bone marrow, and thymus. B cells develop and mature in bone marrow. That's why we call them B cells. So B cells, they develop and mature in bone marrow. That's the cause that we call them B cells. T cells develop and mature in thymus. That's why we call them T cells. So that's the reason that we say T cells. T cells develop and mature in thymus. B cells develop and mature in bone marrow. So bone marrow and thymus are generative or primary lymphoid organs, and peripheral lymphoid organs include lymph nodes, spleen, and mucosal lymphoid tissues and some lymphoid cells present in skin. These are secondary lymphoid organs: lymph nodes, spleen, and skin, and mucosal lymphoid tissues, for example, present in respiratory tract or gastrointestinal tract. They are scattered lymphoid organs in this area. So, what is the structure of a lymph node? This picture shows the cross section of a whole lymph node
Lymph nodes are present in the way of lymphatic vessels. So are lymphatic organs, lymphoid tissues. So we have a vessel that brings lymph to the lymph node, which will be called afferent lymphatic vessel, and a lymph vessel that through which lymphoid cells and lymph exits lymph node, which is called efferent lymphatic vessel. Lymph nodes are composed of two parts: cortex at the periphery of the lymph node and medulla at the center. Cortex is composed of follicles. Follicles are round structures. Here you see are composed of follicles. These round structures that you see are in the cortex. And you see that these follicles have a paler central part. So follicles are in cortex. Lymph node is composed of cortex and medulla. A lymph node, cortex here, and medulla here. Cortex is composed of follicles. You see that most of the cortex have a paler area at the center. These follicles are composed of B cells. And the cells between the follicles is called paracortex, and is composed of T cells. So the immune cells in lymph node are present in the cortex of lymph node. Cortex is composed of follicles and paracortex. The paracortical area is composed of T cells, and the follicles are composed of B cells. Follicles, when they are activated, they have a pale area in the center, which is called germinal center. When we have germinal center, it means that the lymph node has been introduced to antigens, and the follicles have been activated, and germinal centers have developed. And since it is a lymphoid organ, the antigens are brought to the lymph node through lymph. So in this picture you see it's a lymph node. The afferent lymphatic vessel brings dendritic cell, the dendritic cell presents antigen to the lymph node. So for the first time, this dendritic cell has brought an antigen through this afferent lymphatic vessel inside the lymph node. B cells and T cells have entered the lymph node through R3, and they can sense the antigen for the first time. What is the meaning of naive B cell and naive T cell? Naive B cell or T cell is a lymphocyte that has never experienced an antigen. So a naive cell is a cell that has never encountered an antigen, has never experienced an antigen. Then when this naive T or B cell it senses an antigen which is brought by phagocytes or dendritic cells, they become effector cells.
So these T cells or B cells, they can bind antigens for the first time. Then they are called effector cells or memory cells. Some of these B cells or T cells, they play a role in rapid immune responses. They are called effector cells. But some of those cells, they have the duty of having memory. They are called memory cells. They are accumulated somewhere in lymphoid tissues, including bone marrow, and they are activated for the second time when the same antigen enters our body. So these naive cells become effector cells or memory cells. Some of them become effector cells and some of them memory cells. And memory cells, they don't do anything, just wait for entry of the same antigen for the second time. And this picture shows staining of B cells and T cells by immunofluorescence technique. B cells are stained green. These are the follicles. T cells are stained red, present in the space between the follicles, which is called paracortex. So both of them are present in cortex, but follicles are B cells and T cells in the space between the follicles. If you want to have a break time, after the break time, you will